Good evening, everybody. I hope that you can hear me. I'm seeing some people trickle in. My name is Chrissy, and I am with the Friends of Netarts Bay Watershed Estuary Beach and Sea, which we affectionately abbreviate to be WEBS. I'm excited you all are joining us tonight for our second installment of Netarts Bay Clams with Oregon Department of Fish and Wildlife. We are excited to have Dr. Tony D'Andrea back tonight to share some more in-depth information with us. If you missed part one of this series, it's available on Facebook and Instagram for you to view, and you can look at that at any time. We can also send you a link if you are not a fan of using Facebook or Instagram. The first installment went over how to clam in Netarts Bay and the basis of clams, and tonight Tony will take us a little bit deeper into some of the research ODFW is doing in the bays along our Oregon coast and how they're using that information to help them determine how to manage bay fisheries. Before I hand it over to Tony, I'd just like to share a little bit about Friends of Netarts Bay and give you a little bit of background on the logistics of how this will work. We are a Friends of Netarts Bay WEBS is a small nonprofit. We're dedicated to sustaining the Netarts Bay area through education and stewardship. We fundraise to bring free events to our community for both school groups and for the general public. We strive to help people understand this unique area, why it is so special, all of the wonders that exist here. And we think if we are able to do that, then you will be more responsible and good stewards and take care of this beautiful place as you are living here, visiting here, or working here. This is a atypical kind of situation for us. Normally we would be hosting this presentation in person, but unfortunately, because of the way our current situation is, we are doing all of our events virtually right now. The silver lining is that we get to capture this information and we will have it to share as part of our library for the future. And folks can come back and view it again and learn more from it. So we are excited that Tony was willing to do something like this with us virtually. We are going to hand it off to Tony. We're excited to have Tony. Tony comes with a lot of experience doing research in Oregon. He's been with an estuarine ecologist for over 20 years, investigating clam populations on the Oregon coast with Oregon Department of Fish and Wildlife for some time now. Tony comes with a background in academics where he went to school. He's worked with the COAS at Oregon State University and has attended various universities across the country. I think Tony's presentation is going to be really interesting tonight, so I don't want to waste any more time. I'd like to hand it right on over to Tony so he can share with us tonight. Thank you, Tony, for joining us.
Thanks, Chrissy. So good evening. I hope everyone who's virtually attending is safe and healthy. As Chrissy mentioned, in a different year, I would probably be conducting a second in-person clamming clinic in Netarts Bay. So obviously a little bit more challenging this year. So earlier this summer, in discussions with Chrissy Smith and WEBS, we decided to provide information about clams and clamming in Netarts Bay virtually. So this is, as Chrissy mentioned, the second of 3 talks I'll be giving about clams in Netarts Bay. The first talk was a guide to clams and clamming in Oregon. The talk tonight will be on the population distribution of clams in Netarts Bay and how ODFW manages clam fisheries. And then the third talk this fall will be on the ecology of eelgrass in Netarts Bay and their importance to clams and other shellfish. Today, I plan to share with you a brief introduction to the ODFW Seeker project and the Netarts Bay assessment, the methods that we used in our Netarts Bay survey, some results on the bay clam populations of Netarts Bay, examples of how the information generated by this and other seeker surveys can be used to manage estuarine resources. And finally, I'll do a short section on how to find and access seeker information and products, including clamming maps that we produce from our surveys. Before I start the talk today, I just wanted to take a few moments to talk about COVID-19 impacts on recreational clamming. First, I just want to make you aware of some of the closures. One is that recreational clamming is still currently closed to non-Oregon residents. Crabbing, however, is open to all people with a shellfish license, whether resident or non-resident. And also, ODFW offices are currently closed to the public through at least October 31st. Staff are still available via phone and email, and that would usually be the best way to contact anyone. In general, ODFW is asking that sport clammers comply with any state, county, and city closures, avoid traveling long distances to get to a clamming spot on the coast, and practice safe social distancing while out on the tide flats and parking areas. The ODFW shellfish program has been monitoring sport clamming along the coast, and in general, people have been pretty good about safe social distancing, and we certainly appreciate the efforts to date by sport clammers to keep these fisheries open in these challenging times. So the Seeker project, which I run, Seeker is an acronym for the Shellfish and Estuarine Assessment of Coastal Oregon. So Seeker is a permanent project within the Marine Resources Program of ODFW based in Newport. The mandate of Seeker is to generate baseline data for all Oregon estuaries on bay clam populations, track changes in these populations, and inform the public. The main products of the project include stock assessments for bay clams, estuarine habitat maps, tracking non-native species, and public outreach to share the results of the project. The ultimate goal of the project is to provide the best available data for ODFW in the state of Oregon to manage shellfish and estuarine resources. So where has Seeker been to date? Seeker has assessed 6 bays since it became a permanent project and recently completed the survey in Coos Bay in 2019. By 2020, Seeker has data for all of the major bays where both recreational and commercial bay clam harvest occurs. So this includes Tillamook, Netarts, Yaquina, and Coos Bays. These are also the bays where most of the oyster aquaculture occurs in the state, and with the exception of Netarts Bay, are also the most industrialized ports on the outer coast. I wanted to take a moment before I start the talk to introduce what we mean by shellfish assessment within the context of the shellfish program at ODFW. So stock assessments are processes that use different types of information in order to manage and regulate commercial and recreational fisheries. So there are 2 main types of information that we use. The first is fishery dependent data. And so this data collection measures fishery effort either directly from the fishery or indirectly using effort estimates based on interviews, clammer counts, or direct measurement. From this, we can estimate annual harvest by the recreational and commercial fisheries and information on where harvest is taking place, the species composition of that harvest, and the size of clams that are taken in the fishery. In contrast, fishery independent data is data collection that is taken, as the name implies, independent of the fishery and harvesters. The goal is to provide information on the biology of the clam species, distribution of the clam populations and habitat, abundance, and biomass. The easiest way to describe this type of data collection is that it's trying to estimate how many total clams are in a given estuary. So this data combined with an understanding of the biology of the species is compared to the fishery-dependent information to identify whether the harvest levels are sustainable. So the primary role for Seeker within the shellfish program is to contribute to our understanding of the fishery-independent data sources for the bay clam species that are managed by the program. So let's discuss the goals and methods for the Netarts Bay assessment. So Netarts Bay is among the most important bays for clamming and crabbing in the state. The high salinity and large tide flats in the bay provide excellent shellfish habitat. Many of the tide flats are foot accessible, so you don't need a boat to find good clamming spots. The bay is a historically popular area for both clamming and crabbing in the state. There is both commercial harvest of cockles and oyster farming in the bay. And there's been a shellfish preserve protecting a portion of the shellfish populations from harvest since the 1970s. So these varying uses make it important for ODFW to map and quantify estuarine resources in this bay. So there are 7 major bay clams that make up the bulk of clamming effort in the state, which includes both native and non-native clams that are found in Oregon's estuaries. The 4 native bay clams, cockles, butters, native littlenecks, and gapers are the primary targets for sport clammers. However, there are also several non-native species also harvested. They include the purple varnish clam, which has been common in Oregon estuaries since the 2000s. The softshell or eastern softshell, which was introduced in the late 19th, early 20th century. And in Netarts Bay, there's also the manila littleneck. You might recognize the manila littleneck here because it's the species you commonly see in the grocery stores and seafood markets as steamer clams. Most of them are farmed littlenecks from Washington state. However, there is a small natural population that occurs in Netarts Bay. For my talk today, I'll be discussing results for the 4 major native bay clam species. So Netarts Bay was studied by Seeker in 2013 and 2014. In 2013, we focused our effort on the east side of the bay and the shellfish preserve area here in the southern portion of the bay. We also surveyed the major subtidal channel in Netarts Bay, here noted in light blue. We completed our study with the west side of the bay, this large tide flat on the west side of the bay, as well as the area around Happy Camp in 2014. Also noted on this and subsequent maps are the primary public access to clamming flats in the bay by blue stars. So here's one here by Whiskey Creek, up here by the shellfish kiosks. So most of the recreational clamming in general takes place on the east side of the bay. In contrast, most of the commercial intertidal clamming occurs on the west side of the bay. In total, we surveyed around 1,700 acres of intertidal habitat and 150 acres of subtidal habitat. Note that the subtidal habitat comprises less than 1/10 the intertidal area. This is important to keep in mind later on when I talk about the abundance of clams in these 2 different habitats. So our survey was designed to answer 4 questions about clam populations in the bay. First, how does clam population distribution differ among the tide flats and tide levels that occur in the bay? Second, is eelgrass an important habitat for bay clams? There's both a native and non-native eelgrass that occurs in the bay. We wanted to document differences in clam populations among the different eelgrass habitats. Third, are there differences between subtidal and intertidal populations of bay clams? And then finally, are there populations of bay clams in the shellfish preserve? Another way to think about this is whether there are populations that are protected from harvest by commercial and recreational fisheries. In our study, we use 4 types of sampling to address our research questions. We measure tide height on all of the tide flats. We have a method called our rapid assessment method to look at estuarine habitat. We have a method called our detailed assessment method to sample clam populations, and then a dive survey to sample subtidal clam populations and habitat. I'll briefly describe each of these methods and highlight the types of information we get from them to address the major questions about bay clam populations the study was trying to answer. So we measured tide height during the survey in the intertidal and sub areas of the bay. The reason tide height was included in the survey is that tide height matters to intertidal animals such as clams, since it determines the amount of time a clam is submerged in water during the day. As you go down the intertidal beach from the upper shoreline down towards the subtidal areas of the bay, clams spend more time of the day underwater and have more time to feed and breathe. This then affects clam growth and reproduction. This is why when we think of good clamming tides, we think of minus tides that expose the clams found in the lower intertidal areas that are typically more productive than clams found in the higher intertidal. Although this is the typical pattern, there are some clam species that are adapted to living higher up in the intertidal beach. So tide height along the shore can determine where clams are found, and because of this, we explicitly studied this effect on bay clam populations in Netarts Bay. We conducted our rapid assessments to collect the habitat and environmental data along grids shown here with points separated by 100 meters, which is a little bit larger than a football field. This broad survey permitted us to collect habitat information across all of the intertidal area in the bay. At each of the sites noted on the map, we recorded information on the habitat in meter squared quadrats. Within each quadrat, we documented information such as vegetation, such as eelgrass, environmental characteristics, and biological features that might structure clam populations. These data allow us to map the distribution of habitat throughout the bay. In addition, I've overlaid the white points here, which represent our additional random points that we sampled within the bay, and this allowed us to have habitat characteristics on the locations where we also collected clam population data to identify important habitats for the various clam species. So how did we sample clams? While we collected detailed environmental and habitat data at the dam sites, one of the major differences between the RAM and detailed assessment methods is that we extract all clams and other shellfish at the detailed assessment sites. To take these as samples, a barrel was inserted to the sediment to a depth of about 1¼ feet. Using a suction system shown here, we sucked out clams which are captured by a net attached to a suction hose. This method permitted us to capture all clams greater than 1/4 inch in size and to collect juveniles and smaller members of many of the shellfish. And then all those shellfish were measured and weighed. And so during this survey, we collected shellfish at almost 600 locations in the intertidal areas of the bay. The last method we used during the survey was a subtidal dive survey. This is the first study of the clams in the main channels of Netarts Bay in over 40 years. The study of both intertidal and subtidal populations is essential to understand bay clam populations, since bay clams can be found both on the intertidal flats as well as tidal channels, so sampling only one of these gives an incomplete picture of the clam populations of the bay. The other reason the subtidal component is important is that many of the subtidal areas of the estuaries represent a refuge for adult breeding populations called spawning stock. For those in the audience not familiar with clam reproductive biology, clams spawn releasing eggs and sperm into the water column. Fertilized eggs develop into free-swimming larvae that return to the intertidal and subtidal sediments after spending time in the water column growing, thus providing the next generation of clams in the bay. So we sampled 75 sites during the subtidal survey, indicated here by the red dots on the light blue subtidal channel area in the map. The subtidal survey was conducted in coordination with a commercial dive group. These samples were collected using a diver-operated suction end and a water pump operated from the surface. And it was similar to the intertidal system that we used for our detailed assessment sites, and also comparable to the system that ODFW used in the 1970s when they last sampled the subtidal clams. The next section will present the results for the 4 major bay clam species. For each species, we will have a map identical to the one shown here for cockle clams. The map shows both the number of clams found per square meter represented by colored circles. The larger the circle, the more clams that were found. So for cockles, this is 1 to 2, 3 to 5, or greater than 5 cockles per square meter. We sampled almost 600 intertidal sites, as I mentioned, but not every site had a given species of clam. So sites without cockles for example, but sampled in the survey are represented by the black dots here, which you can see. Cockles were found in all of the regions we surveyed, but were most abundant on the west side of the bay. There was on average almost twice as many cockles at a given site on the west side of the bay relative to the east side tide flats. However, there was no difference in the average size of cockles between the east and west sides of the bay. Cockles were present throughout the intertidal, but were more abundant in the mid-intertidal than higher in elevations. So that represents somewhere between 0 to plus 1.6 feet relative to mean low water. Cockles were also associated with eelgrass habitats. As I mentioned before, there are two types of eelgrass present in Netarts Bay. Pictured here on the bottom is the larger native Zostera marina, that forms large dense beds in the bay, and the much smaller non-native eelgrass Zostera japonica, pictured here on the top. In Netarts Bay, there are places where only the native is present, where only the non-native is present, and where they're mixed. We found that cockles were more abundant in habitats where only the native eelgrass was present compared to habitats with non-native eelgrass. Before discussing the subtidal data for cockles, I wanted to describe to you in more detail the spawning stock concept I introduced and what it tells us about clam populations in Netarts Bay. For our purposes today, a spawning stock is defined as the part of the clam population with limited or no recreational or commercial harvests. Thus, these spawning stock populations can supply or enhance the number of clams in areas subject to harvest. This idea has been used to manage fishery resources and is called the precautionary principle. Spawning stocks can be found in the intertidal or subtidal areas. For example, in Netarts Bay, the Shellfish Preserve represents a potential intertidal spawning stock area since neither recreational or commercial clamming is permitted within the bounds of the preserve. Evidence of a spawning stock includes percent of total population biomass protected from harvest and larger and presumably older clams found in the spawning stock areas relative to fished areas. So what were the results for the subtidal? This map shows the density of subtidal cockles represented by the squares, along with the data we've already shown for the intertidal populations. If we compare the intertidal and subtidal habitats, the subtidal population has higher density of cockles relative to the intertidal. At a given spot, you would find more than twice as many cockles in the subtidal portions of the bay relative to the intertidal. This map shows the weight of cockles per square meter in the intertidal and subtidal areas of the bay. Despite only composing 10% of the total area of the bay, the weight of cockles was much greater in the subtidal relative to the intertidal, and this smaller 10% total area had 55% of the total biomass or weight of cockles in the bay. Our second criteria for identification of a spawning stock was larger sizes of clams. Cockles in the subtidal are consistent with this criteria. So this shows relative size of intertidal and subtidal clams. And so we found the greatest number of larger clams and almost all of the clams greater than 3 inches in length in the subtidal channels relative to the intertidal areas sampled. These larger clams were also presumably older, and if so, represent a portion of the cockle population that has had more opportunities to reproduce during their lifetimes. These generally larger clams in the subtidal are part of the reason for the high biomass found in the subtidal, since clam weight scales with length. Thus, both the larger subtidal populations and larger cockles support the idea that the subtidal habitats in Netarts Bay support spawning stocks. As you'll see, this is unique to cockles relative to the other bay clams in the bay. Using our detailed assessment clam data, we're able to estimate total stock biomass of cockles for the bay. These estimates were calculated using the average weight of clams from the detailed assessment samples, scaled by the area of the given tide flat where they occur. When looking at these numbers, keep in mind that biomass incorporates both the number of clams as well as the overall size and weight. For example, if you have two sites with similar densities but differing in average size of the clams, then there can be greater differences in biomass than we would see in densities. Summarized here are biomass estimates for cockles for each intertidal region in pounds. This is really thousands of pounds. Keep in mind that the preserve is the region where no harvest occurs. The east is where most of the recreational harvest occurs, and the western side of the intertidal is where most of the commercial harvesting occurs. Combined, they can be used to estimate total intertidal biomass, which is over 200,000 pounds. As noted in the previous slide, the biomass in the subtidal is higher than the combined intertidal areas, despite only comprising a small area of the bay. Note that for each of these estimates, there are error bars which account for variability in our samples. For each of our estimates, we have a mean biomass estimate represented by the bar. So for cockles, that's this 218,000 pound number. And lower and higher estimates that account for variability and confidence in our estimates. For example, the lower estimate is around 149,000 pounds of cockles, and the upper estimate is a little under 300,000 pounds. And so we expect that the true amount of cockles are somewhere in between that for the intertidal population. Throughout this talk, when I compare the estimated biomass of a clam species with fisheries harvest values, I'll be using only the lower estimate of stock biomass. So everything compared for cockles would only use our most conservative estimate of the biomass of cockles in the bay. So in this case, it would be the 149,000 pound number. So we can compare our stock estimates with data from creel surveys. ODFW has dedicated staff to conduct surveys of clammers, their effort, and their catch. Cockles are the only clam allowed for commercial harvest in Netarts Bay. Shown here are the estimated harvests in thousands of pounds by recreational clammers in orange, by commercial clammers in black, and then the combined total harvest between the two fisheries in white along here for the years of 2008 to 2014. Harvest by both sport and commercial clammers varies by year. Generally, sport harvest has been greater than commercial harvest. However, 2014 had an especially high commercial catch of cockles. We can use the harvest data to look at what proportion of the estimated biomass of cockles in the bay are harvested in the recreational and commercial fisheries. And so what I've done here is overlay the proportion of the total intertidal stock that we determined from our surveys. The lines represent either 10% of the total biomass in the intertidal or 30% of the estimated intertidal biomass of cockles in the survey. Remember that the largest biomass in cockles was found in the subtidal population, so these proportions would be very different if we also included the estimate of biomass for the subtidal population. Even so, the cockle harvest over the 7 years included in our fishery data ranged from 16% to over 40% of the lower estimate of the intertidal cockle biomass in the bay. These numbers are within the range seen for other sustainable bivalve fisheries and are consistent with sustainable harvest based on the age and life history of cockles and the large subtidal population free from harvest. However, the large swings in harvest, particularly from the commercial clam fishery, resulted in action by the ODFW shellfish program for cockle fisheries in the bay. So in response to the seeker survey results from Netarts Bay, as well as an earlier survey in Tillamook Bay, the ODFW shellfish program proposed changes to commercial bay clam fishery regulations, which was approved by the Oregon Fish and Wildlife Commission, which is the state's rulemaking body for fish and wildlife. Three of these rule changes directly applied to Netarts Bay. First, a quota was established on the commercial intertidal harvest of cockles. So the commercial harvesters were limited to 22,000 pounds of cockles per year. And this was based on the 10% of the standing stock biomass of cockles in the bay based on the seeker surveys. In addition, there was an area restriction imposed where the commercial harvest was restricted to a 548-acre section in the western section of the bay, outlined here in pink and blue. And this essentially separated all the recreational effort away from where most of the recreational effort occurred. And then we also installed a minimum size of cockles for the commercial fishery. And so a minimum size of 2.75 inches was established for the commercial harvest of cockles. And if you remember back to the size of the intertidal clams that we found within our survey, that would be less than 5% of the clams that we found on the intertidal flats were greater than this number, thus releasing most of the cockles found in the intertidal areas for the sport fishery. The goals of these rule changes were to both conserve cockle populations and hopefully decrease the conflict between the sport and commercial stakeholders within the bay. So let's switch gears and talk about butter clams. So butter clams were the most abundant clam in the bay. They were present in every region we surveyed. There was a clear difference in size of butter clams between the tide flats on the east and west side of the bay. Butter clams on the west side were on average a ½ inch larger than those on the east side. Butter clams occur throughout the intertidal, including higher tidal elevations. So this makes it so that you do not need an especially low tide to go out and harvest butter clams within the bay. So there's lots of opportunity for clamming for butter clams. And we found that butter clams were also associated with eelgrass, with butter clams more abundant in habitats where the native eelgrass was present relative to habitats with only the non-native eelgrass. In contrast to the results for cockle populations, few butter clams were found in our subtidal surveys. In fact, they were present in only 2 of the 75 sites that we sampled. However, butter clams were abundant within the shellfish preserve in the southern portion of the bay. Approximately 12% of the total biomass of butter clams in the bay were found within this region. And so this portion of the population residing in the preserve may serve as an important intertidal spawning stock for this species. Summarized here are biomass estimates for butter clams for each region in pounds. The totals are for the totals for the intertidal and subtidal are also shown. The majority of the butter clam biomass, about 1.5 million pounds of clams, was found on the west side tide flats. The west side is also a larger area. The small difference in the average size, as well as the larger area of this region, can account for this large disparity in biomass that we found. Recreational harvest of butters relative to our estimate of total biomass in the bay was low for the 7-year period from 2008 to 2014. During this time, recreational harvest averaged less than 2.5% of the total biomass estimated from the seeker surveys. Native little neck clams were very abundant in Netarts Bay. These clams were most abundant in the southern portion of the bay, particularly near the Whiskey Creek area, just north of the Shellfish Preserve. Native little necks are all larger on the western side of the bay, similar to what was found for butter clams, but the difference in size for native little necks is less than the size of a pencil eraser, or about a ¼ inch. So it isn't as big of a disparity as we found for butter clams. Native little necks tend to inhabit higher tidal elevations. This means there's lots of opportunity to clam for little necks, even at moderate low tides. And then finally, native little necks are associated with eelgrass habitats in the same way as butter clams. They're most abundant in habitats with native eelgrass compared to habitats with only non-native eelgrass, although they were found in both of those habitats. Like butter clams, there was a very small subtidal population found in our survey. There was a relatively large population, however, in the shellfish preserves. So 30%, or about a third, of the estimated biomass of native littlenecks in the bay were found within the boundaries of the shellfish preserve. And so this too may be an important intertidal spawning stock area for this species. This slide shows our estimated biomass of native littlenecks in the bay. As noted with the previous slides, the majority of clam biomass for native littlenecks is found in the intertidal areas of the bay, with an estimate of around 500,000 pounds of native littlenecks found within Netarts. For the intertidal areas, native littleneck biomass is second only to butter clams for Netarts Bay. This is really good news for native littlenecks, since many of these populations have been declining in the Pacific Northwest, and in some of the other bays that Seeker has surveyed, we too have seen some declines in or lower densities and biomass in native littlenecks relative to some previous surveys. But based on our surveys, the native littlenecks are thriving in Netarts Bay. The data here shows estimates of recreational harvest of native littlenecks over a 7-year period. Relative to other bay clam species, littleneck harvest is fairly low relative to the amount of clams found in our survey. Note that the scale shows harvest maximum at less than 3,000 pounds per year, relative to tens of thousands of pounds is what we found for most other species. In all years, recreational harvest represents less than 1% of the biomass of littlenecks in the bay. Interestingly, the small harvest does not reflect the availability of native littlenecks in the bay I discussed on the previous slide. So it's really unclear whether this is due to sport clammers' preference for other clam species, that there may not be knowledge as far as where the populations can be found. And probably one of the big players is access, since anyone who has gone after the native littlenecks in the Whiskey Creek area knows that area is a much muddier area, harder to walk on, a lot more work to move around in that area. And the other large populations of native littlenecks, you would need a boat to access that western side of the bay. So finally, we'll talk about gaper clams. This is the major bay clam species found at Happy Camp in the northern portion of the bay. So that's shown here in this inset map. In particular, in this low intertidal cobbly and algae covered area there in the lower intertidal, that supports a really dense population of gapers, including some sites where we found over 50 gapers in a square meter. There are a few gapers found in the shellfish preserve, so this region does not protect any gaper clam spawning stock. And gapers were most abundant in the lower intertidal regions of the bay, so negative low tides are needed to harvest gapers, especially at Happy Camp. If we zoom in on some of these regions, we see that gapers occur in very patchy, small-scale dense beds with surrounding areas having no or few clams. So you can see that in these two inset maps. It's difficult to sample a population when it is clustered and patchy like this, using the random design we used in our large-scale whole estuary surveys. The examples shown in the inset maps are from finer scale sampling of some of the gaper beds in Netarts Bay. Note the high densities and tight clustering that are found within these finer scale sampling that we conducted in this area, relative to the broader distribution of random points in these regions represented by the black dots where no gapers were found. So the random sites often miss the patches, leading to high variability and in general underestimate the gaper population. So another way to look at that is try to think of how to visualize that. And so here's an example of an area of a tide flat with several patches of gaper clams. I'll now randomly sample 4 meters squared to this area a couple of times. So in this case, there are 14 gapers in the area, but in our random sampling, we would only sample 1 to 4 gapers out of the 14 in that area. Thus, the sampling would underestimate the actual population density. One way to get more biological information on gaper clam populations are to conduct focus digs within defined gaper beds. So we did this to complement our random survey. And so these inset maps that I showed you earlier are the results of the targeted digs, along with data from the random samples. Here, the targeted digs do a better job at capturing the fine-scale patchiness of the gapers on both these west side and east side areas on the tide flat, and demonstrate how random sampling by itself misses some of these patterns. That said, because the targeting sampling was specific to gaper beds, they have the opposite problem for random surveys, and they tend to overestimate gaper populations since most of those samples have gaper clams within them. However, this info can still be used to look at some important aspects of gaper populations in the bay, including spatial distribution of the clams and biological measurements such as size and weights of clams. We're currently working on ways to incorporate both approaches in future studies to improve our information and data on gaper populations in Oregon estuaries. Our subtitle surveys found few gaper clams. They were present at only 3 of 75 sites. At one of these sites, there was a very dense population of gaper clams, but pretty much absent elsewhere in the subtitle. So it's likely that they also occur in these patchy distributions of small scale, similar to what we observed in the intertidal regions of the bay. In contrast to other species, there were no regions of the bay for gapers where spawning adult populations were not also subject to harvest. And so they're one of the 4 species, the only one that doesn't have any clear spawning stocks that are protected from harvest. This figure shows our biomass estimates for gaper clams in Netarts Bay from the random data only. You can see the large error bars around our mean estimates due to the patchy distribution of gapers and difficulty in sampling the populations accurately. The range of our estimates could be as low as 38,000 pounds to as high as 2.3 million pounds. Because of this variability, we only compare our mean estimates to the data from our creel surveys of sport harvest. Using the mean estimated biomass, less than 3% of the estimated biomass of gapers was harvested by sport clammers during the years of 2008 to 2014. However, if we were to use the lower estimate of 38,000 pounds as we have for other species, the harvest rate is much higher and can be between 50% to over 200% of the estimated biomass. This disparity further highlights the pressing need to improve our approach to sampling gaper populations in Netarts Bay and other Oregon estuaries to reduce this variability so that our stock biomass estimates can be better used to manage this important clam species. So we're actively testing alternative sampling methods for gaper populations this summer in Yaquina Bay that can be applied to the next Netarts survey, which is currently planned for 2023. There are several ways that seeker data is used to manage shellfish and estuarine resources in Netarts Bay and other Oregon estuaries. So I'll just give you a few examples. Seeker produces clamming brochures based on our surveys. This is one of the ways in which our data can be used to help clammers know where to go when they go out on the bay, or to assist new clammers have a successful and fun first clamming experience. Information collected on our surveys can also be used to produce bay clam stock estimates by estuary to be used by ODFW shellfish resource managers. This information helps ODFW determine whether recreational and commercial use of shellfish resources is sustainable. I showed one example in my talk today on how this type of information is used for managing the commercial cockle fishery in Netarts Bay. Seeker data is also used to track non-native species in estuaries. I mentioned that Netarts Bay has a population of non-native eelgrass. There are also several non-native clam species that occur in Netarts Bay, most notably the purple varnish clam. The presence and distribution of these species are collected during our estuary surveys and can be used to track changes and potential impacts of non-native species on the estuarine ecosystems in Oregon. And finally, information collected during the Seeker surveys are used to track changes in clam populations through time. Seeker surveys of Netarts are planned every 10 years, so we can track changes in estuarine resources. And the current survey that I presented today represents the baseline data for comparison for these future surveys. All Seeker products can be accessed on the Seeker website, shown here. The website has much of the information I talked about today, including our methods, where we survey, our results, clamming maps, and other publications produced by the project. There's also a Seeker dedicated email address. If you have any questions about Seeker or bay clams, feel free to send a message. And then another great resource for clams and clamming in the state is the My ODFW website. And there's a specific crabbing and clamming page on that website that has lots of resource for about these different resources and where to go and techniques for having a successful crabbing and clamming trip. So I want to thank you for your time, and I'd welcome any questions you might have. As Chrissy mentioned, use the chat function in Zoom or the comment section in Facebook Live, and I'll attempt to answer as many questions as I can in the next 5 to 10 minutes.
Thanks, Tony. That was great. We did get a couple questions coming in. One was about actually native versus non-native eelgrass. And I'm just scanning up. So how abundant is non-native eelgrass in the bay, and is there a management plan to remove it?
So there is non-native eelgrass distributed through a large portion of the bay. What we have observed is that when we first did our 2013 and 2014 survey, it was a much larger distribution that had been seen in previous years. But subsequent surveys that we've been conducting since 2016 have actually seen the recovery of the native eelgrass. And currently, we do not have a plan for eradication of non-native eelgrass. There are a lot of species, including recreationally and commercially important shellfish that utilize the non-native eelgrass as habitat. And at least to this point, there is no evidence that there is a negative ecological impact of the non-native eelgrass at this point for the negative effects of potentially spraying herbicides that would have a strong effect on the clam, crab, and other invertebrate communities within the bays that the non-native eelgrass occurs. Although I will say that I'll have a big discussion about native and non-native eelgrass in the next talk in the fall when I talk about eelgrass ecology and shellfish in Netarts Bay.
Great, that was good information. And I'm actually really excited about that talk, Tony. I think it would be really interesting to hear. I've enjoyed talking to you about the eelgrass populations already. Yeah. We do have a comment coming in from Facebook. We have a question about, do you know when the Coos Bay Seeker data will be available from 2018?
I think the plan is to start putting together that report this fall. So sometime in early 2021 would be the report on that.
Okay, early 2021.
Yep.
Great. So some other that came in from Facebook here is, was there any species of clam or crabs that did like the non-native eelgrass? So I think the question is there any species crabs or clams or other invertebrates that prefer non-native over native eelgrass?
I wouldn't necessarily say prefer, but it has expanded habitat for some species into portions of the intertidal that were previously unvegetated, but now have the non-native eelgrass, such that those populations have expanded into those areas and may not have been found in those areas where they're not the non-native eelgrass beds in those locations. And native littlenecks is one of those species that was fairly commonly found within the non-native eelgrass beds.
That's very interesting. So is that for Netarts Bay, or is that for across all bays?
That's for Netarts Bay, mostly because in many of the bays, the distribution of the native and non-native eelgrass are they're spatially separated from each other, where the non-native eelgrass tends to occur in the very high upper portions of the beaches and intertidal flats, and the native eelgrass tends to occur on the very low ends of that. So you don't actually have any interaction between the two species there. And so being high on the upper intertidal, it's also a very different clam and other shellfish composition that you find in that portion of the tide flat. Netarts is actually one of the few locations where you have lots of co-occurrence of both native and non-native eelgrass at the same tide heights on the tide flats.
Interesting. See, we're going to be very excited about this eelgrass presentation. We already have so many questions. Another one that came in via Zoom is, what was the biggest clam you found? And did you eat all the clams you caught during the survey? That's an interesting question. I've never even thought to ask you that before. What do you do with the clams after you harvest them? That's a good question. Anyway,
Yep.
So what's the biggest clam you found, and what do you do with them? Do you eat them?
Okay. Gaper clam was the biggest one, and it was close to 2 pounds. So it's a very big gaper clam. As far as eating clams, we do not eat the clams. We return them to the bay. Usually we'll collect, when we do our surveys, for our detailed assessment surveys, we would collect them one morning, measure them that afternoon, and then return them to the estuary the next day.
Interesting. That's interesting to hear. Must be hard to put some back sometimes, or maybe not if you get them all the time.
Yeah.
Another comment from Facebook is, and I'm not sure exactly which bay, so maybe the commenter can share. But they said that we saw, I'm assuming it's Netarts, we saw a boat with a motor running and a large tube from the boat to a sandbar on the morning of the 13th. Was this a clam survey being conducted?
No. The only work that is being done clam survey wise outside of the razor clam stock assessments on the Clatsop beaches near Astoria are being done currently in Yaquina Bay, and that's all being done by foot and hand tools.
Okay. So what do you think it is that we saw? Was that commercial clamming in progress, or hard to say?
It's hard to say. Well, commercial clamming has to be by hand and hand powered tools, so it can't be anything powered. So it can be rakes or other tools. The only thing that can be used mechanical forms like pumps and other things is if they're actual commercial shrimp harvesters for bait. So harvesting ghost shrimp for sale to bait shops for fishing.
Gotcha. So it sounds like somebody agreed with you online. Probably a sand shrimp harvest. Sounds like you would have been a commercial sand shrimp harvest. And then a question came in. Do you know how the non-native eelgrass was introduced?
I think it was during transport of oysters from Japan for aquaculture. Is the thought that they were that the packing materials was the the Japanese eelgrass, which is native to Asia, as part of the initial shipping over, and that some of the seeds then got introduced in estuaries, and then that was part of how it was transported initially here and then expanded from there.
Yeah, that sounds familiar. I'm looking. So this is a good question, actually. Do individual clams travel much, or do they mostly stay in the same place? And I think you probably have to go by clam on this one, but I thought that was a good question. Yeah, I'll let you answer.
Most of the bay clams do not really move that much. So particularly butter and gaper clams are pretty much in the same place from when they were juvenile clams. The only clam that really does move around quite a bit are cockles. And I'm sure everyone can see the YouTube videos of the cockle clam jumping, using its foot to jump, and it does do that. And I know some colleagues of mine working at the EPA lab here in Newport showed that was a reaction to avoid predation and also avoid poor environmental conditions. So they were trying to jump away from things like seagulls trying to grab them as they were on the sediment surface. But most of the other species of clams live in burrows that are anywhere from an inch to a foot and a half into the sediment, and they do not really move around that much.
I know that I have heard things about cockles like they can move a whole football field's length in a tide cycle. Do you know any? Is there? I've never really been able to confirm any of that kind of information with hard research. I'm not sure if anybody's tagging cockles and finding them the next tidal cycle. But I'm just curious if you had any kind of knowledge of, well, how far would they maybe move if they needed to?
Yeah, I don't have the, from the people I know that worked with mobility and cockles, I don't remember offhand what the how far they moved, but it didn't seem, when they talked about it, was not a huge distance. It was more like meters to tens of meters. But if they're on the surface and the tide's coming in and there's a current, they could easily be moved by the current to another location on the tide flat, which would be greater transport. I know some researchers have proposed that idea for maybe movement of some clams from the intertidal into the subtidal areas by essentially being at the edge of the tide flat and then being rolled off the edge, so to speak, in tidal currents down into the subtidal areas of the bays. But I haven't seen any really good studies that have shown that.
The idea of them getting washed to the other location is not as much fun as thinking of them flipping their foot for a whole football. That is way more entertaining to me. I appreciated some of the data that you were sharing about recreational versus commercial harvest, and we did get some questions about that, actually. And so it was there was a curiosity about why there might have been a spike in recreational harvest earlier. I think, I'm not sure, the early 2009-ish, 10-ish timeframe. And then what was the spike in 2014 that shifted away from recreational to commercial? Do you have a handle on what that was? Why was there a spike? And particularly in cockles, it seemed like there was a spike early on in recreational and then commercial later.
Yeah. We tend to, that 1 year, 2009, we actually used a different sampling method for our creel that year, which seemed to overestimate clamming effort. And so we've tended to leave that 2009 data set out of our analyses because of that. Because every other all the other creel data that we have has been pretty consistent as far as the range of values that we consistently find for harvest in Netarts Bay. And that one year when we used a different method was the one that came out as that spike. And so we think that that was just a problem with the methods used in that year. And as far as the commercial harvest, that was more, if you also saw that figure, they really didn't start to show up until those last 4 or 5 years. And that was just the timing of that intertidal commercial harvest, which started to pick up in 2003 in Tillamook Bay. And then as the number of fishermen grew in doing that, a subset of those fishermen then went to other local bays to see if there were also cockle populations on the intertidal that they could harvest. And so that was really a year where there was a dedicated group that came in and harvested within Netarts Bay. And that was definitely part of, the timing was okay, and that was also a year that we were actively sampling. So we actually saw those harvesters out there, and that since we had then the new data set, that we could immediately then act on making the adjustments to the fishery so that a spike like that would not happen and affect the sustainability of the cockles in the bay.
Thank you. We have another question. Are there other invasive species that have been introduced by the oyster farming?
The only other species that we've seen within the bay is the Atlantic oyster drill. And I forget exactly when that came in, but that was another one that came transported, I think, because that was when, that was earlier when it wasn't Pacific oyster, but when they were trying to introduce East Coast oyster to the West Coast, and that's how that first got here. And that didn't take because the eastern oyster did not grow well in Pacific estuaries in general. But one of the ride-alongs in those was the oyster drill that was common in East Coast estuaries. And so there is a population on the West Coast in some of the major aquaculture bays. Both we found them in Netarts and we found them in Willapa Bay as well. There's a pretty big population there as well. Yeah. But that's the only one that I know of that was either indirectly or directly associated.
Yeah, it'd be my understanding that these introductions would have happened quite a long time ago. You know, it was like 100 so years ago when these things were during like gold rush times, even when oysters were being shipped in from the East Coast, and then
because that was the same thing. Because that's when the eastern soft shell arrived on the West Coast. Was essentially it was shipped across in the trains and attempted to be planted in a bunch of bays along the West Coast, particularly San Francisco to start with, but and they've been documented in Oregon estuaries since like the early 1900s that have been fairly successful, mostly in brackish up estuary areas relative to the main clamming areas where you find most of the clams that we talked about today.
That's interesting. I didn't think about the story of that clam. That's interesting to hear. Where is the next question? There it is. Do the types of clams you discussed today accumulate Dominic? I always like to say it wrong. You know what I'm talking about. Yep. Okay, go for it.
They do not, or they tend to not, and there are a couple reasons for it. One is that the small photosynthetic algae that carry the domoic acid do not do well in estuaries. So as soon as they get out of full strength seawater, their cells burst, so they're not really available as a food resource for the clams to filter. And then many of the bay clams also, there's this process called depuration, where once you take something in, it's how quickly the body can then get rid of that through its natural defense systems. And so some of the species that we know of that have large accumulations of domoic acid, like razor clams and mussels, tend to have a very slow depuration rate, so they accumulate that and keep it in their bodies for a long period of time, whereas bay clams tend to turn over that material very quickly. And so we do include those in our biotoxin testing collaboration we have with the Oregon Department of Agriculture, and it's pretty much been unmeasurable all of the times that we have submitted bay clams, even in portions of the coast where there are extremely high levels of domoic acid in either crabs or razor clams.
Great, good information. It's really interesting to hear that too. I'm not seeing any more questions coming in at the moment. Feel free to type any last minute thoughts in with us. I'll just add some closing remarks while I'm continuing to monitor things here. If we miss something, we will kind of keep an eye out for a couple minutes after we close up shop here and answer things as we can. If something pops in your mind, you can always email Tony directly. The email's right there on the slide that you're seeing on the screen. You can also reach out to us if that's easier, if that's handy for you because you registered with us directly to join today. So either of those things are an option. If something hits you later in the middle of the night, you're just dying to know this thing about clam research. Those things happen to me. So I just want to say that, you know, this event is hosted by the Friends of Netarts Bay Webs, but we're also it's part of the Explore Nature series of outdoor walks, paddles, and hikes, which obviously aren't happening right now because of the state of the world. But we are partnering with them to bring this kind of content to Explore Nature fans and the audience for Explore Nature as well. So if you want to learn more about Explore Nature, you certainly can by going to the website or finding them on social media. Explore Nature Tillamook Coast is how you will find them using that handle. The next thing up for us is next week. Tomorrow actually, we will have a day of virtual tide pool exploration. So it's a full day. You can check back in with us. It'll be interactive. We're focusing on the art and texture of invertebrates and algae that are found in the intertidal. And so it's something to kind of look for when you have breaks from work or you're looking for a distraction from your day. We'll conclude it with a premiere of a pre-recorded presentation by our own marine biologist, Jim Young, at 6 p.m. And then next week, we will shift from talking about clams, which we're focused on this week, to talking about tuna. And so on August 26th, we'll have a presentation by two NOAA scientists on Pacific albacore tuna. And then they will be joined by Amanda Gladix from Oregon State University, who will talk about sourcing albacore tuna by Oregon's local tuna fleet. And then the following day, the Schooner Restaurant and Bar, which did a cooking demo on clams with us last time when Tony presented, will actually do a cooking demo on tuna. So some things to look forward to over the next couple of weeks. Ways to kind of keep your brain active and happy and learning about Oregon's coastal environment and nearshore environment as well. So I just wanted to share those things before we signed off and give people some time for their last minute thoughts. I'm seeing, I do have one more comment come in. That's great. So I'm glad I saw. Do you believe we'll see commercial harvest go to limited entry permits versus open access? Versus open access?
I am not sure. I know it's a topic that the newly formed Tillamook Bay Clam Advisory Committee is going to be discussing over the next year as they are reviewing various aspects of both commercial and recreational clamming in Tillamook County area as part of their responsibility for the North Coast Bay Clam. I'm not sure if that's actually on the agenda. I know it has been brought up in previous years, but never really gained that much traction either from the industry side or within the resource managers at ODFW.
Great. Thanks. I'm seeing some thank yous for your time, Tony. Thank you for the information. It's always nice to hear, though. There's some really good questions that came in both through Facebook and through Zoom. And so it seems like people were really engaged. Shelley, I don't know if you saw anything I didn't see. If there's any last minute thing there, please pipe up.
I think you covered them all. I was about to jump in and then you were on it, so.
I set up 2 monitors for this, so it helped out. It's the way of our life now. No, I think this was really great. It was super wonderful to see the graphs, the data that's coming in, your estimates for what's sustainable out there. Really interesting to see the changes in what your data is showing from commercial harvest and recreational harvest. I was excited to hear about those little necks doing so well in Netarts. That's really interesting and great news. Also, solidified that we have made the right choice to do an eelgrass presentation because clearly there's interest. I was happy to hear that there's no real negative impact from the non-native eelgrass that you've seen. I'm interested in hearing some more about that. Anyway, it was really great. It was a really great presentation, and we're excited to have you back for eelgrass. So we will drum up our questions for eelgrass for sure. So.
Excellent.
It's always fun to have you, Tony. So for everybody who joined us via Zoom, thank you for taking your evening. I hope that you enjoyed it over some dinner or a glass of wine or whatever beverage makes you the happiest. And the same to all of you on Facebook Live. Thank you so much for logging in and your interesting questions and staying engaged with us. We hope you will continue to follow Friends of Netarts Bay Webs and check out some more of our virtual program. And our fingers are crossed that we'll be able to go out and get muddy with Tony next year and actually live out the grand plan that we had planned for this year. It would be really great to have him there and do the cooking demos in real personal, one-on-one way and have these presentations with you being in Netarts. We look forward to that happening. Fingers crossed for next year. So thank you, Tony. Thank you, everyone who joined.
Have a great night.
Yep, have a great night. All right, I'm gonna log us off here.